Hello, am I audible? Good afternoon, everyone. I'm Chitrang from the Chronobiology lab, and today I'm going to talk to you about how circadian rhythms are in the lab versus in nature. So, every day we wake up and go through our day. We eat, we work, and finally we sleep, only to wake up the next day and do it all over again, right? But what is the significance of this? Someone said biological clock, okay. So basically we do these 24-hour or near 24-hour repeating behaviors on a daily basis, and these behaviors are called circadian because the periodicity of these behaviors is approximately 24 hours. And when you are synchronized to an environmental cycle, like the environmental cycles on Earth, it is exactly 24 hours. So these behaviors are not just done because there are some passive environmental cues around you, but because you have a clock inside you that anticipates time and tells you what to do. So what are the functions of this clock? Can you tell? So one function of the clock is to give you information about local time, right? And it has to tell you what is the local time when there is no time cue available. For example, in a situation of constant darkness where you don't have light, constant temperature, etc. Okay, and this property of the clock is ascribed to something that we call precision of the clock, that tells you what is the day-to-day variation in maintaining the period of the clock under constant environments. The other job of the clock is to tell you how much time has passed, okay, and this is happening when the clock is synchronized to the environment. This can be ascribed to a property of the clock that we call accuracy, which tells you what is the day-to-day variation in timing of behavior when the clock is synchronized to the environment. Okay, now this behavior is something that some of you might be familiar with. It's called eclosion behavior, in which adult flies emerge from pupa. This happens once in the lifetime, of course, and this has a rhythm. So more flies emerge during the earlier part of the day, the dawn part of the day, than the rest of the day, and this has a daily pattern, which you can see in this graph. So here what I'm showing you is a light-dark cycle. The dark bars are the night, and the light bars are in white, and you can see a repeating pattern across 4 days of eclosion. So wherever you see these highs, there are more number of flies eclosing during that part of the day. Okay, what we can do is we can summarize these graphs as an average, which I'm showing you here, and the emergence is quantified as percentage here. What you can now look at is different parts of this rhythm. So you can see where the rhythm begins, where it peaks, and where it subsides. So to look closely at this eclosion rhythm in Drosophila, our lab had selection regimes on timing of eclosion to see whether timing of eclosion evolves. And there was selection done on divergent timing of eclosion. So you have this pattern of eclosion. What if you only select flies that are emerging in the morning part of the day or the early part of the day and the evening part of the day separately, and you form populations only of those flies? What you find is after 100 generations of selection or so, there will be divergent emergence rhythms for these early populations and these late populations of flies. Subsequently, we also had selection regimes on how accurate the clock is. So selection was done on accurate timing of eclosion, in which flies that emerged only in a particular window of time were selected, and hence population formed was had subsequent emergence only in that particular window. So this was a 1 hour window. However, these studies are done in the lab, and you see that there are divergent patterns. You see that timing of eclosion can be selected for, but so what? Most of these regimes are in the laboratory with one single time cue, which is light, which is switched on or off. And this is a very simplistic cue, and we know most of what happens to rhythms down to the molecular mechanisms. However, when you go out of the lab, in nature, this is not the case. In nature, there are multiple time cues. There's light, there's temperature, there's humidity, there's food, there's predators, etc. Right? And timing of behavior in nature has to happen with respect to all these time cues. Right? So what about nature is something that we are not able to capture in the laboratory? So to revisit the previous studies by Kaustubh Vaze in the lab, who looked at these early and late populations under semi-natural conditions, which are very similar to natural conditions. So here you have captive flies which are in a natural environment with naturally varying time cues, but food, predation, etc. controlled for. And what you try to see is whether this behavior is repeatable or whether you get a different behavior under semi-natural conditions, which will give you a realistic scenario. What they found was that the timing of eclosion became more divergent under semi-natural conditions. And the early flies became a little earlier, late flies become a little later compared to what they had found in the lab. So here divergence is increasing, right? What Nisha Kannan and colleagues from the lab did was they subjected accurate populations to semi-natural conditions to see whether this phenotype changes in semi-natural conditions compared to lab. And what they found was in semi-natural conditions, the flies became more accurate compared to what behavior they were showing in the lab. Wonderful, isn't it? And they also increased their synchrony under semi-natural conditions. So, does everything become like diverged always under semi-natural conditions? Well, so let's move on to another study in the lab that was done by Priya Prabhakaran and colleagues, which looked at closely related Drosophila species in lab versus semi-natural conditions. What we can see here is there are 3 species of Drosophila, Melanogaster, Malar coetleana and Ananaceae, and these are laboratory conditions. This is when the light comes on. You can see that they are different in their pattern of eclosion rhythm. However, when this study was done under semi-natural conditions, there was no difference seen. So what could this be due to? Differences in the lab might be because there is only light present in the lab, whereas in semi-natural conditions there are other time cues too. So presence of these other time cues here may be causing these species to have similar phenotype. So now, what time cues are really important for timing behavior under semi-natural conditions? So, I mean, I can say that let's ask if Drosophila populations can evolve to utilize these time cues optimally to synchronize with the environment. And well, I mean, anyone will say that of course they can, because circadian clocks are existing in nature currently the way they are because they have evolved to utilize these time cues optimally, right? So, to test this and to find out which time cues might be important, we had 4 large outbred populations called the T24 populations in the lab, from where we derived 4 replicate populations called the NT24 populations, which have now been maintained under semi-natural conditions in an outdoor enclosure for about 118 generations, which is close to 7 years. So these populations were started by a student called Radhika Shinde in the lab, and I'm continuing this. And we want to see how circadian clocks of flies reared in semi-natural conditions have evolved to become different from the ones that are their ancestors in the lab. So firstly we asked whether these 2 sets of populations, which are like say parent or offspring kind of thing, they differ in eclosion rhythm in the lab itself or not. And what we found was that in the lab there is no difference. So if you compare the orange T24 controls with the green NT24 ones, they are overlapping, right? There is no difference even after maintaining them in outdoor conditions for so many years. So what happens when you see the eclosion rhythm in semi-natural conditions, which are the rearing regime for NT24s? So if you look at what environmental conditions look like outside in terms of just light, temperature and humidity, this is a graph showing you something. So on the x-axis is local time, and what you can see in yellow is a light profile across 24 hours. This is temperature that increases with light increase, and humidity decreases across the day. So this is something from November 2017, and now I'll show you how these profiles differ across months. So this is February, this is April, and you can see that it is more or less same, but there are subtle changes if you compare across months, and that gives you the differences which can be classified as harsh for the fly or moderate for the fly. So you can see that June and August had more humidity, and so they were relatively moderate seasons. This is October, January, and what we found was overall from this that in moderate semi-natural conditions, which were humid, rainy, and quite suitable, not very stressful for flies, these NT24 populations and T24 populations did not really differ in their eclosion rhythms. However, in harsh semi-natural conditions, be it hot conditions or cold and dry conditions, these flies of NT24s had a significantly advanced eclosion rhythm compared to the control lab ones. So what in terms of the time cues may be bringing about the difference which allows the NT24s to emerge earlier in such harsh conditions compared to T24s is an interesting question to ask. So to give you some more information about NT24 populations, we've also found that in activity rest rhythms, NT24 populations show higher precision in the onset of activity rest, and under harsh summer conditions, NT24 populations have higher fecundity than T24, resulting in higher offspring number. Higher fecundity means higher number of eggs laid. So what have we learned from this? We've learned that phenotypic difference in the lab may or may not be observed in semi-natural environments. Also, phasing of behavior varies across seasons, which, okay. But what next is more interesting, because we will learn what specific aspects of light, temperature, and humidity cause this difference in phasing of NT24s. We will also want to ask what changes in the circadian clock machinery result in this difference. Thank you.